Hello everybody, this is D. Rajashekar, lecturer in botany, Srimati Kanganeni Kalyani Government Degree College, Vinikonda, Guntur district. In this video lecture, you are going to learn electron transport system and oxidative phosphorylation. What is it actually? In the previous videos, we have come across the two phases of aerobic respiration. They are glycolysis and Krebs cycle. Glycolysis releases two pyruvic acids at its end. These two pyruvic molecules independently get changed into acetyl coenzyme molecules and enter into the mitochondrial matrix where they start Krebs cycle. So that Krebs cycle takes place twice. Because of this glycolysis and Krebs cycle in the aerobic respiration, energy is released in the form of NADH2, FADH2 and ATP. ATP is the main energy biochemical molecule, that is the main form of energy that is present in the living system. Whereas NADH2 and FADH2 are reduced forms, they must be oxidized into NAD and FAD respectively so that they can give ATP, the direct utilizing form of energy by the living systems. Thus, the NADH2 and FADH2 that are formed out of glycolysis and Krebs cycle must be oxidized by a process called electron transport system and oxidative phosphorylation. This is the third phase and the last phase of aerobic respiration. In this video lecture, you are going to learn these two things. Actually, they are not two different things. They are interconnected. One way electrons are getting transported through complexes or enzymes. So, the ATP is generated in the presence of oxygen. That is why it is called electron transport system and oxidative phosphorylation. Oxidative phosphorylation means formation of ATP from NADH2 and FADH2 in the presence of oxygen. Before learning it in detail, where do these electron transport system and oxidative phosphorylation take place? I have told you clearly in the beginning itself, glycolysis takes place outside the mitochondria, that is cytosol, and the Krebs cycle is in the matrix of mitochondria, and this electron transport system and oxidative phosphorylation together take place by the complexes or enzymes which are present embedded in the inner mitochondrial membrane. We know mitochondria is a double membrane structure. The inner membrane shows some infoldings called cristae. In these inner membrane foldings, the complexes are present embeddedly. These complexes carry out electron transportation and also involve in the hydrogen ion or protons transfer from matrix to perichondrial space, so that finally we will get the ATP from it. Before going into the details of the actual process of electron transport system and oxidative phosphorylation, first and foremost we must learn different complexes or enzymes which are getting involved in these processes. There are actually 5 different complexes present in the inner mitochondrial membrane, and 2 mobile carriers are also taking an active part in it. First of all, we must know what are those 5 complexes. Complex 1, this is an enzyme, that is NADH dehydrogenase. Its reaction center is ferrous sulfur. And then complex 2, this is succinic dehydrogenase enzyme. Its reaction center is also ferrous sulfur. And the complex 3, this is called cytochrome C reductase. Actually, it contains cytochrome B and cytochrome C1, and its reaction center is also ferrous sulfur. And the complex 4, it is cytochrome C oxidase enzyme. It contains cytochrome A1 and cytochrome A3. It is closely associated with the oxygen molecule, that is terminal electron acceptor. Its reaction center is 2 copper molecules. And the fifth one is complex 5, that is ATP synthase enzyme. It contains 2 parts. One is head region or F1 region. It is present freely in the matrix of mitochondria. And the second part is tail region, that is also called F0 region. It is fixed in the inner mitochondrial membrane. These are the details of 5 different complexes involving in this process. And there are 2 mobile carriers. They always mobile or show movements between these complexes. They are ubiquinone or UQ and cytochrome C. Generally, ubiquinone carrier is showing movements between complex 1, complex 3, similarly complex 2 and complex 3. Whereas cytochrome C shows its movement, it moves between the complex 3 and complex 4. Here we can see all those complexes and mobile carriers. This pink one is shown as 1, Roman number 1, that is complex 1. And here light skin color structure is representing complex 2, that is shown as 2 in Roman numbers. And you can see that light blue color structure, that is complex 3, and sky color structure, that is complex 4. And here finally lavender color structure represents what? ATP synthase enzyme or complex 5. In between the complex 1, 2 and 3, there is a yellow color structure, that is ubiquinone, a mobile carrier. Similarly, between the complex 3 and complex 4, you can see a ball like green color structure, that is representing cytochrome C. So thus, after learning different complexes and 2 mobile carriers, now we have to focus on the actual process. If you see the slide clearly, you can see the below region is representing the matrix part of mitochondria, that is the inner core of the mitochondria. Whereas all the complexes are present in the inner mitochondrial membrane. You know that there are 2 membranes, outer membrane and inner membrane. Within the inner membrane, you can see the complexes present embeddedly, and the 2 mobile carriers are running freely. And in between the 2 membranes, you can see perichondrial space or intermembrane space. And the outer one is called outer membrane. So, the inner mitochondrial membrane is the major part. It differentiates inner matrix of mitochondria and outer perichondrial space. You must remember that clearly. After knowing this, what happens first? Here, the process can be studied in 2 different heads. First of all, the NADH2, the reduced form of NAD, which was formed in glycolysis and Krebs cycle, must be oxidized into NAD plus H+. When it is oxidized, automatically 2 electrons are released, which are given to complex 1, from where these 2 electrons are carried to complex 3 through ubiquinone mobile carrier. There is no involvement of complex 2. From 1 to 3 electrons are moved. From complex 3 to these 2 electrons are moved to complex 4, by cytochrome C. Finally, these 2 electrons are given to oxygen. That blue color line that is shown is representing the electrons movement from complex 1 to complex 3, from complex 3 to complex 4, and finally to oxygen. As electrons are given to oxygen finally, this is called terminal oxidation, or oxygen can be called as terminal electron acceptor. The blue color line represents clearly the electron passage. Meanwhile, at the same time, when these electrons are getting transported from one complex to another complex through mobile carriers and finally to oxygen, proton motive force is developed. When this proton motive force is developed, automatically the hydrogens or protons present in the mitochondrial matrix are entered into the perichondrial space or intermembrane space through complex 1, complex 3 and complex 4. These three complexes are also called proton pumps. See, here you can see by a pink color arrow mark. From complex 1, 4 hydrogens or protons are transported from matrix to the perichondrial space. Similarly, you can see another pink arrow mark, straight arrow mark, where 4 proton molecules or ions are transported from complex 3 to through mitochondrial matrix to the perichondrial space through complex 3. Similarly, you can see the transportation of proton molecules or hydrogen ions from matrix to perichondrial space through complex 4. But the difference here is, here in complex through complex 1, 4 protons are transported. By complex 3, 4 protons are transported. But by complex 4, only 2 protons are transported from matrix to perichondrial space. Why this difference? Here, at complex 4, 2 hydrogens are transported from matrix to perichondrial space. The remaining 2 hydrogens are joined with the oxygen, so that oxygen is changed into water molecule. That is why here only we can expect 2 hydrogens. Thus, when NADH2 is oxidized into NAD, electron transported from complex 1 to 3, 3 to 4, 4 to oxygen through 2 mobile carriers. Meanwhile, proton motive force is developed. By this, 10 protons are transported from mitochondrial matrix to perichondrial space through complex 1, complex 3 and complex 4. Okay, is it clear? When these protons are transported from matrix perichondrial space, a gradient is developed, a difference is developed between the protons of perichondrial space and protons present in matrix. Generally, membrane space contains more protons than that of the matrix. To overcome this gradient, what happens? All these 10 protons present in the perichondrial space or intermembrane space come back to matrix through the complex 5, that is ATP synthase enzyme. See, you can see at the end of this slide, on the right side of the slide. So the purple color structure, a lavender color structure, that is a ATP synthase enzyme, where tail region is tightly fixed in the inner membrane of mitochondria, and head region is revolving in the matrix. As the head region, that is F1, is revolving, like a wheel in the matrix region, it is also called as universal motor. Universal motor. So in ATP synthase, the head region, that is F1 region, is also called universally rotatory motion. Why it is called so? The 10 protons present in the perichondrial space come back to the matrix through ATP synthase through its tail and head region. When 3 protons come into the matrix region of mitochondria, 1 ATP is generated. Thus, how many protons are there? 10 protons. So, how many ATP can be generated? 3 ATP, because 3 protons is equal to 1 ATP, so that 9 protons is equal to 3 ATP. 1 hydrogen is, it is not utilized actually. Thus, 1 NADH is equal to 3 ATP. This is the first phase. And the second phase of the process, here in Krebs cycle, FADH2 is formed. When this FADH2 reduced form is oxidized, similarly as of before, 2 electrons are generated, which are transported from complex 2 to complex 3 via ubiquinone mobile carrier, from complex 3 to complex 4 via cytochrome C mobile carrier, and finally these two electrons reach the oxygen. That is called terminal oxidation. Thus, during oxidation of FADH2 into FAD, electrons 2 are transported from complex 2 to complex 3, complex 3 to complex 4, finally to the oxygen. There is no involvement of complex 1 in this process. Meanwhile, at two different places, hydrogen ions or protons are transported from mitochondrial matrix to the perichondrial space, just like before, because of proton motive force. Which complexes are involving in the transportation of protons here in the second phase? Only complex 3 and complex 4. Don't think of complex 1. So through complex 3, 4 protons are transported, and as of before process, through complex 4, only 2 protons are released from mitochondrial matrix to the space region. So so that what happens? Proton gradient is developed between the matrix and the perichondrial space. To overcome that, again these 6 protons present in the perichondrial space come back to the matrix through ATP synthase enzyme. When these 6 protons are coming back to the matrix region, 2 ATP are generated. As I told you, 3 hydrogens is equal to 1 ATP. So that 6 hydrogens is equal to ATP. That is why in the beginning I told you 1 FADH2 is equal to always equal to 2 ATP. Thus, NADH2 and FADH2 are reduced, the reduced forms are oxidized into NAD and FAD respectively, so that 2 electrons are released, they are getting transported through complexes, and finally they are given to oxygen. Meanwhile, proton motive force is developed, so that protons are transported through these complexes from mitochondrial matrix to perichondrial space. To overcome the proton gradients between these two spaces, again the protons come back to matrix through ATP synthase enzyme. So it was explained by Peter Mitchell. Hence it is also called Peter Mitchell chemiosmotic theory. Thus, no formation of new ATP here. You may misunderstand this actually. There is no liberation of ATP. What happened here? Only the energy present in NADH2 and FADH2, which were formed in glycolysis and Krebs cycle, give the energy in the form of ATP. That's all. So in aerobic respiration, we can expect the formation of energy molecules only during glycolysis process and Krebs cycle. In electron transport system, there is no liberation or formation of ATP directly, but the ATP present in the NADH2 and FADH2 are is released in the form of ATP through this electron transport system and oxidative phosphorylation. I hope you have understood the total process of aerobic respiration clearly. We'll meet in another video lecture. Till then, bye. Have a nice time. Thank you.